leading beyond chemistry.
Guten Morgen aus Essen.
Very
Mein
good
Name ist
morning from
Matthias
Essen.
Ruch.
My name
Ich
is Matthias
begrüße
Ruch,
Sie und
and I
euch
would
alle
like to
herzlich
welcome
zu
you
unserer
to
heutigen
our Research
F&E-Pressekonferenz.
and Development Press Conference.
F&E heißt Forschung und Entwicklung.
Research and
Darum geht
development
es heute.
is
Es
the
geht
topic of today's
Innovationen
press
der
conference.
Zukunft.
It's
Es geht
about
die
innovations
Produkte,
for the future.
mit denen wir in
It
den
is
kommenden
about
Jahren und
products
Jahrzehnten
which we will
wachsen wollen.
use for our growth
Mit
in
mir
the
im
years and
Moment stehen
decades
hier
ahead.
Harald Schwager
Harald
und
Schwager
Ralph Marquardt,
and Ralph Marquardt
die
are with
zunächst
me on
Ihnen
the
in
panel
einem
here,
Vortrag
and they
einen
will
Überblick
start
über
by
unsere
giving you
Forschungs- und
an overview
Entwicklungsaktivitäten
of
geben
our
werden
research and development
und ein bisschen den
activities,
strategischen
and
Hintergrund
will
beleuchten.
also kind of
Im
explain
Anschluss
our
werden wir dann
strategic
3
background.
Fachvorträge hören,
After that,
die
we
sich zu
will
3
hear
spannenden
3
Themen
special
darauf
presentations,
beziehen,
and
wie
they
wir
will
hoffen,
relate
da
to
in
very
den
specific
kommenden Jahren wieder mal
topics
Produkte zu
that
entwickeln.
are to
Wir sind
help
noch nicht
us
so
in our
weit. Es
product
geht heute
development
um
in the years
F&E.
ahead.
Es geht nicht
We are
darum,
not that far yet.
Produkte
Today,
vorzustellen,
it is
die
about
schon am
research and
Markt sind.
development. It is not
Es ist
about
das 6. Mal,
presenting
dass wir
products
diese
that are already available
F&E-Pressekonferenz
in the
machen.
market. It
Es
is for
ist
the
unsere
6th
größte
time
und
that we are
bedeutendste
holding this press
Pressekonferenz, die Evonik
conference
im Jahr
here,
macht.
and it is the
Wir machen
most
die alle
important
2
one
Jahre etwa
Evonik
und
is
ich bin sehr
holding
froh, dass Sie
per
alle
year.
dabei sind.
We are having this press
Schon
conference
während der
every second year,
Vorträge haben Sie die
and I'm happy to
Möglichkeit,
have you
im
here.
Chat Fragen
During the
einzustellen.
presentations,
Die
you
Fragen werde
can
ich dann an Herrn
pose your
Schwager, an Herrn
questions
Marquardt
in the
und
chat,
an unsere
and
3
I will
Referenten
forward
weitergeben.
these questions to Mr. Schwager and Mr. Marquardt and
Das
to the
ist es
3
eigentlich schon
speakers.
fürs Erste. Ich
So much from my side.
begrüße Sie alle herzlich
So
und
once
begrüße
again, I
auch
would
die
like
Damen und
to
Herren hier im
welcome
Saal.
you. I would like
Und
to
ich richte
welcome the
meinen
ladies and
ersten Dank
gentlemen
wie immer
here
an die
in the room.
Übersetzer,
And I would like
die
to
dafür
thank first of
sorgen,
all
dass wir
the
diese
interpreters
Pressekonferenz
who make
simultan
sure
auf
that
Deutsch
we have
und auf Englisch
simultaneous
durchführen.
translation in English
Herzlich
and
willkommen,
German.
Herr Schwager, Sie haben
Welcome, Mr. Schwager, you have the floor.
Thank you very much. A wonderful good morning, ladies and gentlemen, here in the room or outside in front of the monitors and on the cameras. We've brought along fascinating topics. I would like to welcome you most warmly to this year's R&D conference. We have a lot of interesting, fascinating topics. You may still remember our old slogan, and it is the research spirit for new developments continues to live. It's still alive, and we intend to launch many new products tomorrow in the markets, and what this could be is something that you're going to learn about at this press conference today. Evonik has for 5 years been on the road to become one of the best specialty chemicals companies in the world. In the last 5 years, we've made good progress, and with great consistency, we have been integrating sustainability into all the elements of our group strategy. That means, in concrete terms, into the portfolio management, into innovation, into corporate culture. But this does not only go for the integration of sustainability into the group strategy, it is also true of a level lower. In the regional strategies, the specific strategies, this integration has taken place by now. For our transformation, we've been investing in a targeted manner and on a large scale into green growth, and we are turning sustainability into the key driver of our innovation and of our research and development activities. In other words, ladies and gentlemen, we are building the next generation Evonik. This means that by 2030, we will have invested more than 3 billion euros in next-generation solutions. That is, those products that have a superior sustainability benefit. This includes, for example, solutions from the areas of food packaging, but also insulating materials or membranes for gas filtration. We support our customers in making their own products more sustainable and in improving their climate balance, their CO2 footprint. You know, we are the enablers that make things possible and better. We are not the wiseacres. Next generation solutions already account for about 37% of our portfolio, and in the years ahead we intend to increase this to over 50%. Innovation is an absolutely essential element of our strategy. The solutions for a sustainable future can only be found with innovations, can only be found with intensive research and development. And this is, of course, expensive. This requires investments. Therefore, some figures. 464 million euros were spent in 2021 on research and development. This is about 3% R&D ratio. And here we should also mention that 464 is the number in the past few years. That was in spite of the crisis. So this amount has been increased. And when we look to the future, we do not intend to actually slow this down in any way. The largest portion of our R&D expenditure goes into the research activities of the growth divisions, specialty additives, nutrition and care, and smart materials. The division performance materials is focused on process and product optimization. We are focusing on projects that aim to develop solutions and technologies of the next generation. Sustainability in this context is completely integrated into the decision-making processes of our innovation organization. For our innovation projects, we use the same system with which we assess the sustainability of our business. We make sure in this fashion that innovations provide sustainably benefit for our customers. We look at a large number of environmental factors with the aim of reducing the footprint of our products in the environment. And here we are guided by our aim of reducing the CO2 footprint or water consumption. With our strategic R&D, we are placing a clear focus on identified growth fields. We intend to create new additional business that is driven by innovation. Since 2016, our innovation growth fields have been contributing disproportionately high share to our growth. Last year, we could follow on from the successful development. A major contribution in 2021 was made by our innovations around drug delivery technologies, that is the transport of medical agents into the human body. Some of you, ladies and gentlemen, that have been with us for quite some time are familiar with this. In my first paper, 4 of these bars were missing, the ones from 2018 to 2021. At the time, the billion in 2025 was far away. Now, 4 years later, we are seeing that we've made major steps forward, because in 2021 we are now beyond the 500 million, and this is an increase in a very difficult year by 40% from 2020 to 2021. You can see we are on a good road towards achieving the 1 billion. We intend to follow on and continue the successes from the innovation growth fields in the years ahead. And we are working already on topics of tomorrow and the day after tomorrow. The long-term perspective is taken up by Creavis. Creavis has for more than 20 years our strategic innovation unit. Now we have positioned it very clearly as an incubator in order to develop transformative and sustainable businesses that actually go beyond the current product and market focus of the operating business units with a view to the business and applications. The new projects are pooled in Creavis new growth areas, and we intend to reach commercialization within 5 to 10 years. It's about the key topics for the future, for instance, for a hydrogen and circular economy, for environment-friendly and more efficient agriculture, and you're going to hear more about this in the specific presentations that are due to follow. We will provide you with a good overview. We expect here and sales potential in the new growth fields that would be similar to the innovation growth fields. Every growth area should have a potential of about 250 million euros. Not all of these areas will survive. Some of them will be all the more successful. However, this is normal in this risk-prone area of research and development. Of course, we cooperate with relevant universities, industrial companies, but also startups. When it comes to startups, talking about startups, our Evonik venture capital unit is important and comes into play because Evonik venture capital is also a key element of our sustainability and innovation strategy. With a capital expenditure volume of 250 million, we have since 2012 taken shares in now 50 startup companies and specific funds. Beyond this, we have created a new fund with a focus on sustainability. It is focused exclusively on investments in technologies for climate neutrality. The Evonik Sustainability Tech Fund has an investment volume of 150 million euros. You can see it on the slide. It pursues the aim of achieving CO2 reduction by reducing or elimination of emissions or carbon binding. The focus is on the circular economy, alternative base materials, future mobility solutions, hydrogen economy, renewable carbon, and the generation of clean energy. In the next 5 years, 15 to 20 investments are supposed to be made on a global basis. You can see our innovations, whether in the short, medium to long term, are contributing to a sustainable future. Our strategy is on track. We will enable tailor-made solutions for our customers and therefore for the improvement of the lives of people worldwide. We want to continue this path consistently with a lot of commitment, and this is also the key area of a man that is standing left to me. He is Ralph Marquardt, our new Chief Innovation Officer at Evonik, and he holds a PhD in chemistry and has been in the company since 2006 and has held various management positions. Mr. Marquardt, I wish you all the best in your new role. It's your turn now. I'm looking forward to my new role, and I'm grateful for the trust placed in me. Sustainability, innovation, and business interests are interconnected more than ever before today. I consider it to be a personal challenge to find new solutions for a healthy future worth living in. Sustainability is a very personal issue to me. The chemical industry, and hence Evonik, can make a major contribution to this. The chemical industry is in for a major transformation. In the medium term, we will replace fossil raw materials by sustainable materials like plants and bio-waste, or by recycling processes or CO2. We have to change our way of production. We have to consume less energy. We have to become more efficient and have to embark on new roads here. This is a major challenge, maybe the biggest challenge for the chemical industry in the last 50 years. We at Evonik have embarked on our new journey, and we have developed concrete solutions that contribute to a sustainable future. For this, I would like to present 3 examples today. The first example comes from the growth field of Creavis, namely the circular economy. The establishment of circular economy systems is a key building block on the road to climate neutrality. Evonik can play a key role in this by enabling circular material streams. A good example is our new polyurethane recycling process for mattresses. The average useful life of a mattress is about 10 years, and this means that in the EU, about 40 million of mattresses are disposed of every year, with an equivalent of about 300,000 tons of polyurethane. What if we could recycle all these materials? For this, we've developed a proprietary recycling process at Creavis. It comes from comfort and insulation and process engineering at Creavis. The recycling process is clearly superior to the usual processes because almost all the base materials can be recovered. The process provides polyols of a similar quality like original raw materials, and with this we can produce more sustainable soft PU foam or flexible PU foam products. We can actually out-license this or operate this process ourselves. We are in talks with various partners, like customers or PU component manufacturers, and we think about how we can actually establish this new process in the value chain. The second example comes from the Creavis growth field of the hydrogen economy. In the energy transition, major hopes are placed on green hydrogen. First of all, as a future energy source, as fuel for traffic and transport, but also as important raw material for the chemicals industry. But green hydrogen is still more expensive than fossil hydrogen. In order to overcome this, we need sufficiently low-cost electricity from renewable energies, fast expansion of the necessary hydrogen network infrastructure of pipelines on the basis of political outline conditions, and we need efficient electrolysis technology. And for this, Evonik has developed a new anion exchange membrane, AEM for short, that is a breakthrough for the electrolytic generation of hydrogen. With the Durayon membrane, we now have a solution for the cost-efficient production of green hydrogen. Electrolysis with the AEM technology has clear advantages over the established processes, which leads to low investment costs and low operating costs. On the one hand, we make it possible to achieve high current densities, dynamic operation depending on the availability of green energy, and the hydrogen production under pressure, up to 30 bar, and the cheap cell materials can be used, no titanium, no platinum, and in the electrodes, no iridium. With this, we have the leading AEM technology. The production is currently being built up at our Creavis in a continuous production process and is being scaled, and first sales are expected as early as at the beginning of next year. Let us move on to the third example, the Creavis growth field biomaterials for medical applications. It's about collagen. Collagen is a protein that occurs in bones, muscles, skin and tendons, and it forms a framework that provides strength and structure. It is used, for instance, in cosmetics and medical technology, and usually collagen is obtained from animals, but the animal-based collagen is subject to quality fluctuations, which makes it more difficult to process. We have developed a new fermentation process in which microorganisms produce collagen. We've been marketing this under the name of Mycolan. The Vecolan, the high-purity Vecolan, is produced under controlled conditions and is characterized by a consistently high quality. It does not contain any animal components. It is safe, sustainable, and can be scaled in manufacturing. It enables the development of new medical applications in the area of renewable medicine, tissue engineering, aesthetic dermatology, or also in dental applications. I hope I could provide you with the first insight into our fascinating developments in the area of specialty chemicals. I've learned in my career so far, building networks, getting to know various perspectives and looking for dialogue. All of this is essential in order to make good ideas great. And here I'm looking forward to the exchange with you on concrete innovation projects and our ideas for a sustainable, climate-friendly future. Thank you very much for your attention.
Lieber Herr Schwager, lieber Herr Marquardt.
Dear Mr. Schwager, dear Mr. Marquardt, thank you very much for this introduction and these good examples. If there are any questions, dear participants, please feel free to actually post them in the chat. While you are doing this, I use the opportunity and would like to introduce Elisabeth Gorman to you. He's going to be the first speaker of the three technical presentations. She's going to talk about lithium. Lithium is used in drive batteries in many electric cars, in millions of electric cars. The problem is that once it has been used, it's the end, and 95% of them are disposed of as waste. But lithium is a valuable material, and therefore researchers of Evonik look for ways in order to recycle this lithium and to keep it in the economic cycle. How this can be made in the future, on this we are going to hear Elisabeth Gorman.
Frau Gorman, kommen Sie ruhig in die Mitte hier.
Join
Sie
us
sehen
in the
auf dem
middle,
Bildschirm
Mrs. Gorman.
die Charts, die unsere
And
Teilnehmer
on the
aussehen.
screen, you can see
Guten
the
Morgen.
slides that our participants will see as well. Welcome to you and a very good morning.
Frisches Lithium aus
Well,
verbrauchten
fresh
Batterien.
lithium from used
Die
batteries.
Elektromobilität wird kommen. Die
Electromobility is in the
Anforderungen an
pipeline.
das Recycling von
Requirements to the
Altbatterien
recycling
steigt
of used
stark an.
batteries are
Immer
ever
noch wird ein
increasing.
Großteil
A
an
major
Lithium,
part
welches in
of
Altbatterien
lithium,
verbaut
which
ist,
is used
heutzutage
in
nicht
old batteries,
wiederverwertet und
is
landet
not
ungenutzt
recycled
in der
yet
Entsorgung.
and ends
Dies
up in
wollen wir ändern
landfills
mit
or
einem
wherever.
Projekt bei der
This is something we
Creavis.
want to
Im
change within
Labormaßstab
Creavis.
arbeiten
We
wir auf einer
have a laboratory
elektrochemischen
project. We are
Lithiumseparation
working
mittels
towards
Einsatz einer
electrochemical
speziellen
lithium
keramischen
separation
Membran,
by using a
bei dem
special
man Lithium
ceramic
effektiv,
membrane
nachhaltig und
in which
kosteneffizient
lithium
abtrennen kann.
can be separated in a most efficient, also
Schauen wir uns nun die
cost-efficient way.
Dynamik des
Let us
Lithium-Recyclings
look at
an.
the dynamics of
Hauptgetrieben durch
lithium
den schnell
recycling.
wachsenden Markt
This is
in der
mainly driven by the
Elektromobilität.
fast-growing market in
Im letzten Jahr wurden knapp
electromobility.
7
Last year,
Millionen
roughly 7
Elektroautos
million
weltweit
electric
verkauft.
vehicles were
Bis
sold worldwide
2030
until
erwartet man
2030.
hier eine
The
Verzehnfachung.
expectation
Damit
is
wächst
a
der
multiplication
Markt
by
an
10.
gebrauchten
This means that the
Lithium-Ionen-Batterien
market, when it
und
comes
auch
to
der
used
Anteil an
lithium
Altbatterien
batteries is
wächst sehr
growing
stark
and
an.
the share of
Die
old
typische
batteries is growing
Lebensdauer einer
fast as well. The typical service life of a lithium-ion battery, which is integrated in electric vehicles, is typically 10 to 15 years, depending on the driving mode and mileage of the car. We expect a fast growth in volumes of used batteries in the decades to come. The legislator, internationally and European-wide, strengthens requirements to the recycling of lithium batteries. And this leads to a situation where the industry is obliged to search for new and sustainable solutions for the recycling of lithium batteries. Let us look at the lithium market in general. What you see here are volumes expressed in lithium carbonate equivalents. This year we expect 500 to 600 kilotons of lithium carbonate equivalents which are reported, and they are mainly used in the electromobility sector. Until 2030 you can expect triple the number, and until 2050 we will talk about 5,000 kilotons. As you can see here, lithium is a fast-growing market. In violet, you see the share from secondary produced lithium battery from battery recycling of used batteries, other sources, or production waste, battery production waste, as it were. Until 2030, the share will be roughly 10%. But in the decades to come, the share will significantly increase. It could cover 1/4 of the total needs. So the importance of recycling will be growing. What does a lithium cell look like? And where would you find lithium-ion cells? Lithium is contained in cathode material and in the electrolyte as lithium salt. The main cost share of a lithium battery is accounted for by the cathode material. This is the most valuable part for the recycler as well. In the middle, you can see the percentages for the weight of a typically integrated cathode material with the most precious material. Cobalt 4%, nickel 13%, and lithium 3%. And the result is the distribution of the weight. Cobalt and nickel have high output and recovery rates and can be reintroduced into the circular economy. Lithium only has a weight percentage of 3%, and this is not the case here yet. Why is that so? Existing processes which are being used for recycling are so cost-intensive that the recyclers do not recognize any good business model here in order to make sure that the secondary lithium salts, in comparison to the primary salts, could be resold. What you see here is the development of lithium prices of the past years. You see a rapid growth this year with 8 times higher lithium price. We are talking about 70 to 80 US dollars per kilogram lithium carbonate equivalents. This is a typical graph from the spot price market segment. And you clearly see, if you compare that to the previous years with a market value or sales value of below $10 a kilo, that we have experienced extreme price increases triggered by the extreme demand for raw materials. This concerns salts like lithium hydroxide and lithium carbonate. These are the most important salts which are used in lithium-ion batteries as raw material. The legislator is more stringent now too, as far as requirements to the recycling rate in lithium. This is an example of the EU battery directive. At the moment, less than 10% of the lithium contained in batteries is used. As of 2026, this is to increase to 35%, and as of 2030 to 70%. So this is the recycling rate of lithium that is aimed at. The industry is obliged to look for better and more efficient solutions and processes for lithium separation. If you look at a typical life cycle of a lithium salt in the electromobility sector, you will see on the right-hand side at 12 o'clock we will start with the battery raw material, lithium hydroxide and lithium carbonate, which is a primary raw material source. Then the cathode material producer will build it in. Then the entire lithium cell will be the next step. And after a service life of 10 to 15 years as an average, these batteries, due to the lack of remaining capacity, will have to be replaced. They might end up in secondary use, battery storages, and sooner or later they will end up as end-of-life batteries with the recycler. The battery recycling industry is intensively looking for new cost-intensive technologies for the recovery of lithium salts, and the market potential we can see is around 190 kilotons, which is 10% of the total needs. And if you correlate that with the extreme price increase for lithium, you will see that there is a rather attractive market potential. What does the conventional process for the recycling of lithium salt look like? These stems mainly from the primary lithium production technologies. On the left-hand side, lithium solution from the recycling process, where there's a gradual segregation process, additives like sodium tetrachloride, calcium hydroxide, lithium hydroxide salt is then again isolated. This process requires a lot of precipitation process steps, refreshments, chemical reagents, and high level of maintenance and HR investment. The salt included is normally not sufficiently pure to be used in a battery. There has to be a purification process in order to end up with a battery-grade salt. The whole process, therefore, is not really sustainable, but it's cost-intensive. We chose a different approach. We want to recover lithium ion by way of electrochemical membrane processes with a single-stage process. On the left-hand side, we are starting again with a lithium solution from the recycling process, and we will enter that into our electrochemical membrane process. As an end product, we will isolate highly pure hydroxide monohydrate, which can be used as new cathode material in a lithium battery. This process it can be a continuous one, a sustainable one. It's highly efficient when it comes to recovery, and there is hardly any output loss. The core of the electrochemical membrane segregation process is this special ceramic membrane. The ceramic membrane is conductive exclusively for lithium ion. Evonik has got the competences in particulate design and powder production with ceramic membranes. We must design and produce the specific membrane and also integrate it in the module configuration. The innovation has broad competence also in electrochemical processes. Let us look into the structure of the process. On the left-hand side, you see the setup of the electrochemical cell. Basically, we've got a two-chamber system. On the left-hand side, the anode chamber, where the waste lithium water is introduced with all its impurities and contaminations. And it's then separated by lithium selective ceramic membrane, entering into the cathode chamber where water is filled in and where you will isolate the product. Then we've got the ceramic membrane. It's an Li selective conductive membrane which contains a specific crystal grid where lithium, by putting it under voltage, can migrate through the crystal walls. It is only lithium migrating through these separating walls. All the other atoms with the different radiums will be blocked. It means that exclusively lithium will end up at the cathode side, and with the hydroxide ion, they can link up to lithium hydroxide. Our membrane has got a high level of lithium selectivity, high current efficiency and density, and we were able to prove that in a laboratory test. Per transported electron, 1 lithium ion will be transported, and there is a high stability to water and high temperatures as well. This ceramic membrane does not allow penetration of water. It's electrically insulated as well. Now, this is a result of first laboratory tests we did, and it shows the excellent lithium selectivity and service life of the membrane. A typical diagram that shows how in the laboratory we run the electrochemical cells. Service life of almost 2,000 hours is plotted here. That relates to the first membrane system we tested. For the technical implementation, of course, we will have a considerably longer service life we focus on. On the violet line, you can see the increase of lithium hydroxide concentration in the cathode chamber, and the gray line is the separation of lithium ions in the wastewater area. On the right-hand side, this is a bigger flat membrane, and this is where we proved that successively the lithium succession in the product chamber can be increased up to a level which is at the precipitation level of lithium hydroxide. And this has got advantages for the later separation of lithium hydroxide as solid matter and for further processing for battery material. Lithium hydroxide, which you isolate here, and has been proven to have a high level of purity. Let me summarize. The advantages of this technology: we've got a process here that has been developed by Evonik on a laboratory scale, which is characterized by high current efficiency. It is a single-stage process. It does not require any additional precipitation chemicals and can be run as a continuous process. It's got excellent lithium recovery rates of more than 99%, a very high membrane selectivity, and also low water consumption. All that leads to a situation where you can talk about a very sustainable process, and also relating to the CO2 footprint in comparison to conventional processes. This is something that is very positive. We provide for a pressure model in the future which can be an easy plug and play solution to be connected to existing recycling systems and can thus be applied in reality. Our timeline, 2019 was the year where this idea was created by way of an internal competition, and it was actually the winner idea. In 2020, it was successfully implemented in a first feasibility study. Since 2021, it's been an official development project within Evonik. At the moment, our focus is placed on scalability, the draft of a prototype, and we're talking a lot to external partner companies in order to take into consideration and test real wastewater systems. And for the next years, the scalability and upscaling of our process is to be pushed ahead. Thank you very much for your attention. Dear Mrs. Gorman, thank you very much for your presentation. I've got the impression when I look around that everybody knows how things are working. So let me look around. Are there any spontaneous questions to Mrs. Gorman? This doesn't seem to be the case. We haven't received any question in the chat, but please stay with us for the later question and answer sessions. I would like to transfer from electromobility to agriculture.
A major leap, and with this, Jan Wolter is coming. He is working in Farm to Fork at Creavis, and today he is going to talk about how bacteria can contribute to actually supply cereals with atmospheric nitrogen or something like that. So, Mr. Wolter, good morning, warm welcome. The floor is yours. Good morning. Thank you very much. I have a fascinating story to tell you how we can make agriculture more sustainable. And with this in mind, I would like to ask you, how can a growing world population be fed? This is one of the most important questions that we have to ask ourselves time and again. The answer is often similar or the same. We have to step up agriculture. That means more use of pesticides, more use of fungicides, and in particular, large amounts of fertilizers are needed in order to safeguard crop yields and to increase them. So this approach is not sustainable and is accelerating climate change. And this leads on to various challenges for agriculture in Europe. On the one hand, we have to actually increase the productivity of agriculture by about 20%. If the food products that are needed and the animal feed for feeding the world population are supposed to be provided and supplied. The second challenge is climate change. Climate change leads to harvest losses. We have an increasing number of extreme weather events. We have changing growth conditions, and all of this puts plants under abiotic stress, heat, the droughts, salinity of soils, of frost. All of this is happening more often. In addition, society and politics are urging for a transformation of agriculture into sustainable agriculture. This can be seen from the Farm to Fork strategy as part of the Green Deal of the European Union, where we've said that by 2030 the amount of pesticides is supposed to be reduced by half, by 50%. In addition, the amount of fertilizers used are supposed to be reduced by 20%. If you look at the problems of agriculture, you will inevitably get to the point where you have to address the issue of nitrogen. Nitrogen is the driver of plant growth. It's an important nutrient for plants. It's an essential part of proteins and therefore the precondition for plant growth. And in addition, it is the growth limiting factor for plants. The current need, today's need of nitrogen in agriculture is mainly covered by synthetic fertilizers. Over 100 million tons of nitrogen fertilizers are produced globally, and this is more than half of all fertilizers that are used. If you use such large amounts of nitrogen fertilizers, this creates problems. You can see that fertilizers are spread over large areas, but this nitrogen fertilizer that is being spread does not really reach the plants. Only half of it reaches the plants because the roots of the plants, not the entire fertilized, not all the fertilized area can be reached. Over 50% of the nitrogen remains left in the soil and is actually transported to groundwater, surface water by rain. So it's washed out, but the part remaining in the soil is transformed, converted by bacteria into nitrous oxide, laughing gas. Laughing gas is a gas that is 300 times more harmful for to the climate than CO2. In addition, fertilizer production, which is one of the most energy-intensive processes we know, 1.2 tons of CO2 are produced in order to produce 1 ton of fertilizer. And fertilizers are in short supply. Natural gas is used as raw material and as energy source in addition. One of the great hopes in agriculture are biostimulants. Biostimulants improve the soil. Biostimulants increase the root surface area of plants, and in addition it helps to withstand abiotic stress that is on the increase due to climate change. Furthermore, biostimulants can actually facilitate the uptake and assimilation of nutrients for plants. This then enables us at this point to reduce the use of chemical fertilizers, synthetic fertilizers, and unlike fertilizers, biostimulants don't have their own nutrient content. So you cannot actually provide overdoses of fertilizers, and therefore they promote growth, they safeguard the yield, the crop yield, and they are environment friendly. Our idea was to provide the plants with nitrogen from the atmosphere and with bacteria. And if this idea is successful, this makes a major contribution to sustainability by actually significantly reducing the nitrogen fertilizer. This is known, this process is known from leguminous plants in their roots, and we actually translated this and transferred this to wheat and corn. We have actually selected these plants, these crops, because they can use this more efficiently than when it comes to soil applications. In our environment, we always have air. This is the same for plants and for humans, and in this area we find 80% of nitrogen. The plant can unfortunately not utilize and assimilate this nitrogen, but plants breathe day and night like humans in order to assimilate, in order to adjust to environmental conditions, and in order to exchange gases. The main constituent of the air, nitrogen, cannot be used at this point in time by the plants. And here our microorganisms come into play. If the air, by opening, it penetrates the leaf from underneath through openings, it gets in contact with our microorganisms. These microorganisms can transfer the nitrogen from the air into ammonium, and ammonium is a form of nitrogen that can be assimilated very well by the plants. Through pathways, this is carried into the plant and is then available for plant growth as a building block. In order to get to such a kind of microorganism, we actually searched for plants in extreme habitats, plants that grow on soils that are dry and poor in nutrients, that are contaminated with heavy metals, because if plants can actually grow under these conditions, they of course have a feature, a characteristic that actually distinguishes them from other plants. Therefore, we analyzed them from microorganisms and bacteria, and we isolated them, especially those responsible for the growth. And then this unique collection of bacteria that has been obtained from this has actually been exposed to stress, to shortages of nitrogen, and we watched which ones of these bacteria grow nonetheless, because if they grow, this is an indicator that they master the quality of fixation of nitrogen. We then determined the bacteria specifically by DNA sequencing. This enables us to look at a later time what future potential is hidden in these bacteria. Microorganisms and bacteria on their own have a very short lifetime if they are removed from their normal habitat and are carried to a new environment. At this point, the agri-science of Evonik comes into play, because we are dealing with chemical formulations. Our microorganism has been combined with the silica combinate in order to come up with a unique Evonik formulation solution. This is characterized by cypernate, of course. It is in the carrier fluid breakthrough, and here it is stored in a stable manner on the cypernate. Our microorganisms have been deposited. This combination has been covered by a water-soluble protective layer, and then a product has been produced in this manner that can be stored. If this product is actually applied in practice, you just have to mix it with water. In this moment, the bacteria are released, and the farmer can use the product with the normal spraying equipment in order to apply it to the crops. And here the breakthrough unfolds its true effect as a spreader. This spreader makes sure that the fluid is spread across the entire surface of the leaf, not only on the upside but also underneath on the bottom, and here the organism reaches the stomata and can enter the leaf in order to unfold its full effect. All of this has been tested in greenhouses, in greenhouse trials, and we demonstrated that the principle works. On crop plants, on corn plants, we have been able to demonstrate that with half of the fertilizer use, the same size of crops has been achieved, but these crops have more than 30% more root surface area, and this actually makes sure that they're much more resistant to the effects of climate change. At present, we are in the process of demonstrating the effectiveness in fields. In the next few months, we will harvest the first crops, and then afterwards the formulation will be optimized and fine-tuned, and we expect to launch it in the market in 3 to 5 years from now. Our contribution to agriculture at this point is the optimization of growth and health of crops, of plants, efficient application with the existing equipment, so farmers do not have to make any new investments, long storage life of the biostimulants, and compliance with future legislation requirements, so best possible crop yields with less fertilizer. Our contribution to the agriculture and society is the reduction of the use of nitrogen fertilizers. By the reduction of the use of fertilizers, we are also reducing the greenhouse effect. We strengthen plants and make them more resistant to the effects of climate change, and we provide an ecological contribution to safeguarding world food supply, and we make the sustainable agriculture
Herzlichen Dank.
realistic? Thank you very much, Mr. Wolter, for your presentation. Let me look around again. Are there any spontaneous questions
Fragen an Herrn Wolter.
on what you've just heard? Any questions to Mr. Wolter? This is not the case. Then I would like to actually
Versichern, dass wir all Ihre Fragen
reassure you
aufgenommen
that
haben. Wir
we
werden
have
nach dem
actually
Vortrag
recorded all the
Fragen
questions. After the third presentation, we're going to have a round of questions, and we'll come back to Mrs. Gorman and Lithium. Now I'd like to introduce someone to you who I don't have to introduce to you, because those that have been with Evonik for quite some time are familiar with Walter Pfefferle.
Ein sogenannter Senior
He's one of our most experienced researchers, a so-called senior fellow, and he will also look a little bit into the topic of nutrition, but we are moving from wheat towards meat. And you know that Evonik has a special relationship with chickens, and that brings us to epigenetics. If you don't understand what you're saying, has never understood Mr. Pfefferle, but we have got the opportunity to listen to him and get to know what epigenetic means and why it is so important for nutrition in the future, and for the question, what can we call good meat? Mr. Pfefferle, you have the floor. Thank you very much for this very kind introduction, and I do hope that I will manage to make come true what you said. Well, the chicken that is telling its own story. No, you still see a human being in front of you, so we have not totally turned into something different yet. But I will take you on a journey, and I will tell you how we can come into a situation that a chicken will be able to tell its own story sooner or later. Now, the way we deal with food will change. And I think we just want to have fairness in everything. So we assume that when we eat something, it is good for our health, and we also want to make sure that all that stuff is sustainable. So a lot has changed already. Now, when we look at the global situation, we see that this will not be easy. I think at the moment we are roughly 8 billion people on the planet. This year we will probably exceed the 8 billion limit. And what you see here is 8.5 for 2030, so the number is increasing. And then there's another phenomenon. Globally seen, life standard is still rising. Per capita, we have to expect that we will still have higher meat consumption. And that means, on the other hand, that per person there is less and less surface available. Fair, healthy, sustainable. That's not easy. How do we deal with that? Now, when you look at the different types of meat people consume, you see that in the meantime, chicken meat has become the most dominant meat. So it has kind of replaced pork in the past few years. So it's outwinked, so to speak, pork. And there's some reasons that explain it. For chicken, for poultry in general, there are no religious taboos. And we know that keeping chicken is relatively simple, less laborious than with other animals. And at the end, with poultry meat, we've got an extremely significant resource efficiency we can achieve. You just need 1.6 kilograms, depending on how you proceed, feed per kilogram of meat you want to produce. This is much less when you think of pork. Then I think it would be 2.6. With beef, it would be more than 6.5. So the share of poultry is already dominating today, and it will continue to grow. So that is why we want to look into the production of chicken meat. When we look back, you see that in science there were decades where they looked into the individual disciplines. So there was the discipline or the faculty of animal nutrition, then animal health, then there was livestock farming, then we see that these components have merged. we consider all these ingredients to be one single system today. And what has to be added? We've got the concept of types of livestock farming. So there's an additional component, namely the animal welfare has become a kind of point of attention. That's an interesting thought, isn't it? When we talk about animal welfare, we are not really talking about management systems where the human being is monitoring and controlling things. No, we are kind of putting an animal's glasses on. And this is our approach. This is the future. This is where we want to get it. And this is where we will say, we will make sure that the chicken is able to tell its own story. Sounds simple. Once again, a simple sentence here in the title. Life leaves its mark on us. And I think everybody would say that's true. I'm a biologist, and these simple sentences, as I learned during university studies, are extremely difficult to translate into science. And in order to explain that, we'll just take a journey into biology. Just a tiny excursion. Genes, you all know what genes are. The totality of genes is a genome. So that's what we call it as biologists. And my genome is my sphere of potential. It kind of defines me, or how and what I can be, what I can turn into. So that was the left-hand side of the chart, and let us move towards the right-hand side. You see the phenotypes. The phenotype, young, old, sporty person, less sportive, written by sorrows, etc. So you see, our way of life and what we've experienced puts marks on it. And don't forget, with an identical genome, we are never changing genomes in our life. So how can you explain that correlation? And there is a discipline in biology which is called epigenetics. And epigenetics is the discipline that is explaining the different phenotypes. Biologists are talking about the phenotypical determination. A step further, an essential mechanism of epigenetic approaches is the DNA methylation. As a matter of fact, our DNA is changed at certain places. A methyl group is attached to someone else. And this methylation can be understood as instruction for regulation of genetic activity. The methylation has an influence on the question as to whether gene sequences will be switched off, on, or upgraded or downgraded. And now I have to add something else. Very frequently used gene sequences are subject to less methylation. So I've got a kind of feedback mechanism. The way I'm living is mapped here. So the life does change the instruction to the genome. That's so exciting. And obviously, this leads to certain methylation patterns. And these methylation patterns can be interpreted. This is roughly the principle behind what I'm going to tell you now. Now, once again, something very exciting here. Now, when you look at the methylation patterns of a human being and look at them at certain age groups and collect them and put them in a certain order, you will need a lot of computational capacity. And then you will see the methylation pattern changes with age in a certain way. And on the basis of the methylation pattern, we can present the biological age in comparison to the chronological age. The epigenetic clock, this is the way we call it, will now tell us the true age. the way I'm feeling, the biological age, as it were. And why am I telling you that? Now, our idea was that methylation, epigenetic or biological clock could be created for chicken. So wanting to do something is always the beginning, and then you'll find it's not that easy to implement it. The challenges were tremendous. Just imagine a genome of a chicken has got roughly 20 million different spots of methylation. These methylation sites have to be identified. Nobody has ever done it. Nobody has ever been interested in that biological clock for chicken. The data you retrieve have to be processed in a quantitative way. Each time, each sample, 20 million sites, etc. And then in research, the situation is that I can only generate knowledge if and when I work with material which has been precisely defined. Otherwise, I cannot create any correlation. We needed explicit samples, and also we needed a kind of reading technology which was reliable and accurate. And at the very end, you have to be able to cope with these tremendous volumes of data. You have to be able to assess and evaluate them in order to draw conclusions. We are talking about data volumes here of more than 10 terabyte. So tremendous amounts of data. How did we proceed? So, approach is, if you cannot cope with the situation alone, you look for people who can help you. So we're really happy that we were able to get Professor Lücker from the German Cancer Research Center Heidelberg on board. He is such an experienced expert who for many, many years, more than a decade, has been working on epigenetics. So he's got a very deep biological expertise. And then I said the reading technology is very important. We got together with the technology leader in this area, the Illumina company. And the third component is the fact that you need your own teams as well in order to make sure that what you want to generate can be translated in an application in areas which are of interest to us, namely application in animal husbandry, for example, in chicken. And we managed to do so. Well, otherwise I wouldn't be standing here and I wouldn't be presenting this topic today. We managed to create an epigenetic clock for the chicken. On the left-hand side, what you can see here is in green the different factors which decelerate age. In red, you see factors which accelerate aging. Sounds familiar, doesn't it? So between chicken and us, there's no major difference in that way, at least. Well, free range translating into the human domain would be sports, could be helpful on the green side. On the right side, the red side, I will focus a little bit more. These are the categories, pathogens we can expect. Disease, for example, poor quality feed, poor ranging or husbandry conditions, and stress. So some say there is positive stress, but biology knows stress which will create or result in diseases and accelerate aging. On the right-hand side, you see, well, that is the outcome of the 10 terabytes plotted here in a system of coordinates. On the one hand, on the x-axis, you've got the chronological age. On the other side, you've got the biological age. In a normal situation, you would see that this is correlating. We could say, if the chicken has a normal aging process, we'll see a direct correlation between the biological age and what you can actually read in the birth certificate of the chicken. But then there are deviations. We see data points. What you see here is animal welfare low, where you would expect that the biological age is lower than what you identified. And this is the situation where there was no well-being and where somebody could see under laboratory conditions that there was a disease, that the chicken was ill. And this led us to a new concept, the derivation of a health status on the basis of this process is absolutely possible. Health status is more or less identical with animal welfare. From our point of view, this is a very exciting and fantastic result, namely that with the help of such a test, we were able to make things transparent. Now, what is the current status? So, we have developed a special epigenetic test. On the right-hand side, you see how we use it, how we can use it. We've got a piece of meat. We can take samples. We can process the sample. We put them into a measuring device. We can read data. And we can interpret the data. So this is up and running. That whole process is existing. The technology as such has been set up and tested by us. This is what we're doing in Singapore. And we have also set up our own team there. Two, actually. Two groups. One is looking into epigenetics, biology, and the other one is exclusively dealing with bioinformatics. This is the process of evaluation of these huge volumes of data. So what we call it is generating knowledge from just seeing some data. This is a separate discipline. And these groups can handle the technology and can also explore new fields of application. Now, the summary. This is exactly what you saw in the title at the beginning. The chicken is now able to tell its own story. And what is that story about? It's about how the chicken was brought up. And it's quite nice to see that this story becomes accessible and becomes accessible in the whole value chain. And thus it becomes transparent. Once again, a very nice title. Well, more or less, you can't contradict, can you? Even when you look at your private environment, you will always confirm that transparency generates trust, won't you? And I think this is a crucial element of what we are doing. Where does that transparency come from? What you see here as a major part in this poultry chain, we have the upbringing, the feeding, then we've got the poultry producer. And here, the poultry producer, if that person invests in good keeping, in expensive feed, whatever they can do better, they can prove now what they have done well or better. And they can show it to everybody. In the retail, you would talk about quality. The origin and quality of meat can be proven. This is one element which was not existing in the past. And then on the consumer side, they can see the data. They obtain reliable information on what they are eating. So transparency generates trust, especially if and when transparency is based on solid scientific results. What is our offering now? It's a new epigenetic test for chicken meat. It's scientifically based, it's easy to handle, it's affordable. And who are the addressees of this test? Now, the proof is meant for those in the poultry value chain. Certifiers, producers, as I just mentioned, retail traders, and also for the hotel and restaurant business, for example. What kind of tests are available already? I was talking about the epigenetic clock, the methylation clock, which tells you something about animal welfare and the health status. amongst others.
Das ist auch etwas ganz
And
Besonderes. Wir haben
there's
Nachweise
something
über die
special. We've
Verwendung
got
von
proof
Antibiotika.
about the use of antibiotics.
Wir haben Nachweise über die
We
geografische
can
Herkunft.
prove the geographic
Und
origin.
wir haben auch
And
Nachweise, wie
we
das
can
Tier
also
geschlachtet wurde.
prove the way
alles
the
geht mit dieser
chicken
Technologie.
has been killed. All that is
Mit
possible
dieser Technologie
by
ist es
way
nun
of
so,
technology.
dass Evonik, die ja schon
Now,
eine ganze Reihe von
Evonik
Produkten, die
has
anfangs
already
eingeführt,
launched
im
a
Bereich
series of
Tierernährung
products
hat,
in the
dass wir
area of
da
animal
jetzt einen
nutrition, animal
weiteren
feed,
Block dazufügen,
has now added
eine weitere
another block,
Systemlösung zur
another system
Nachhaltigkeit.
solution, which is meant for sustainable meat production.
Was kommt jetzt?
What's next?
Wir haben entwickelt, wir haben validiert.
We have
Jetzt
developed
reden
the
wir
whole
mit den
thing, we've
interessierten
validated
Parteien.
it, and now we are talking to
Wir reden
interested
mit denen,
parties.
Wie unser
We're
Test einsetzbar ist,
Talking about
ob
the
es noch
use of our
Justierungen
test,
braucht.
whether there's need for
Wir können
adjustments.
relativ leicht, weil wir ja die Technologie
Since
komplett im Griff haben,
we've
leicht
coped with the
weitere
whole technology,
Qualitätsnachweise
we can
nachentwickeln.
easily
Die
develop
Verfügbarkeit
additional
der
quality
Lösungen,
proofs.
so wie vorgestellt,
The availability of the
das ist
solutions
kurz- bis mittelfristig der Fall. Wir sind
can be
also da.
implemented at mid and
Fazit.
short-term basis, so we are up and
Ich
ready.
denke,
Conclusion.
verspreche nicht zu viel, dass
I
wir
don't
da
think it's
ein neues
promising too
Kapitel
much when I
aufschlagen.
say that we are opening a new chapter.
Ich möchte sagen, dass
I
eine
would say
nachhaltige
A sustainable
Geflügelfleischproduktion
poultry
jetzt greifbar wird, die
production
explizit das
is
Tierwohl
now
miteinbezieht.
available,
Und der Verbraucher
taking
bekommt
into
damit
account the animal
Transparenz
welfare.
auf dem
Consumers
Teller über
are
das,
given
was er braucht.
transparency about the food on
Vielen
their
Dank für die
plates.
Aufmerksamkeit.
Thank you very much for your attention.
Lieber Herr Pfefferle, vielen Dank für Ihren Vortrag.
Mr. Pfefferle, thank
Als
you very much for your
erstes, bevor ich
presentation.
weiterspreche,
Now,
möchte ich
before I
Ihnen ein
continue,
Kompliment
I would like to
weitergeben, das
make a
während
compliment.
Ihres
This is
Vortrags
something we
eingegangen
received
ist
during the presentation.
von Herrn
Mr. Where is it?
Kaiser.
Where is he?
Hör mal. Das gebe ich Ihnen dann gleich weiter, wenn ich es
No,
wieder gefunden habe. Hier ist
I
im Moment
can't
einiges eingegangen.
find it.
Es sind auch
I've
zwei
received so
Fragen,
many
die Sie
comments, but there are two
eingegangen. Die ich als
questions
erstes
we
Ihnen stellen
received as
möchte.
well,
Ich
which
würde
I would
dann
like to
danach
ask. And
die beiden
after
anderen
that, I would
vielleicht
like
noch mal nach
to
vorne zu bitten,
ask the two
weil auch
other
für Sie
speakers to
Fragen
come in
eingegangen
front
sind.
once again, because we have
Zum
received
Ende
questions
dann
for you
Herrn
as well.
Schwager und
And
unseren
at the
neuen
end, Mr. Schwager and
F&E-Chef
our
noch mal
new
nach
research and
vorne
development
Nun zunächst
manager
eine
will
Frage
come
für
forward
Herrn
as well.
Pfefferle
First of all, a
von
question to
Philip
Mr. Philip.
Der
Philip
fragt,
Broadwood.
does the
He's asking,
insight from the research inform or influence the ability to produce lab-grown chicken meat without animals? Also bekommen wir die Möglichkeit, Hühnerfleisch sozusagen in Zukunft ohne Hühner?
chicken meat without chicken, can that be generated in future in laboratory, for example?
Die Möglichkeit ist auch
Well,
heute schon da.
we do have the possibility
Die Frage
already
ist,
today.
bei all diesen Dingen,
Now,
wie
with all
nachhaltig
these
sind die
things, we have to
einzelnen
ask the
Lösungen.
question about sustainability.
Ich
How
weiß, dass es in
sustainable are the
Singapur
individual
jetzt
solutions?
soweit ist, dass
I
diese
know that in
Lösungen
Singapore
auch,
they are
kommerziell
quite developed
werden.
already. They are coming
Wenn
up
wir
with commercial
darüber
solutions.
reden,
Now,
welches
when we are
Ausmaß
talking
kann das
about
annehmen,
the
dann müssen wir ja
dimension
auch
it
die
can take, then
Nachhaltigkeitsfragen
we
für
have
diese
to
Produktionsform
also ask about
stellen.
sustainability in the
Da
production
bin ich
process.
skeptisch, wenn
And
man
I'm
die
rather skeptical
Ökobilanz macht über das
when you
gesamte
set up
System,
an eco
ob
balance
das
over the whole system,
dem
across the whole
Globus,
system.
gut
I'm
wird.
rather doubtful
Wenn
as
man
to
jetzt
whether
nur
it
die
is
ethische
good for the globe.
Bewertung
If you go
in den
for an
Vordergrund stellt und
ethical
sagt, das
evaluation
überschreibt alles
only,
andere, dann
and if
mag
you say
es
it
ein
overrides
Bereich
all the
sein,
rest, it
wo
might
man
be an area
erfolgreich Dinge
where
an den
you
Markt
could
bringen kann.
successfully
Wenn man das
launch
jetzt, wir hatten vorhin über
products in the
8
market.
Milliarden,
We talk
8,5
about
Milliarden Menschen
8.5
geredet. Wenn man
billion
sich
people
vorstellt,
on the
was
planet.
man dafür
If you
Anlagen
imagine
braucht, dann ist das ein gewaltiges
what you
Ausmaß.
need, then
Da
it
bin
would
ich ein
be
bisschen skeptisch. Also ich würde sagen, für
tremendous
Nischenbereiche
dimension.
ja.
I would say for
Eine weitere Frage
niches
aus
it
dem
would be
Stream, Herr
possible.
Pfefferle.
Another
Sind
question
noch
from the
weitere
stream.
Anwendungsbereiche geplant, die
Do you
über
plan
das
other
Nachhaltige
application
hinausgehen?
cases going beyond
Womöglich für andere
chicken
Nutztiere?
meat, maybe
Also
for other
wir
animals or other types of
haben
meat?
natürlich darüber nachgedacht
Yes,
und
we
wollten zunächst einmal
thought about it,
mit einer
but we
Spezies
wanted to start off
die
with
anderen
one
Dinge, die
type.
werden nachkommen.
All the
Man
rest
muss
will
einfach
follow.
schauen. Also wir werden lernen müssen.
Oh, we'll have
Ich
to
habe Ihnen
learn
gesagt,
anyway.
die jetzigen
And I
Schritte
did tell
sind,
you
mit dem
the
Markt zu
current
reden, was wir denn
steps
diese
are to
genauen,
talk to the market and see what kind of application areas you will go for from the point of view of the value chain. And once we know that, we will draw conclusions and use the lessons learned and maybe apply it to other animals as well. And then I found the comment I wanted to transfer. Georg Cape from Transcript said, absolute highlight today, epigenetic presentation. So he kind of felt in university, and he's asking a question. The types of slaughtering, how can you prove it epigenetically? That is, from the biological point of view, it's relatively easy. Now, I'm the philosopher now, and I hope that everything I'm saying is scientifically verifiable. The types of slaughter which we differentiate are those which show different types of bleeding behavior, so to speak. And since epigenetics can differentiate between different types of cells, we see that in a certain method of slaughtering, if blood is retained in the tissue to a larger extent, we could see it. So due to the fact that it is a very comprehensive technology that covers all types of cells, we can look at the remaining blood in the body of the dead animal and can thus differentiate. We can clearly differentiate signatures, and that would be the biological reason. Now, I promise you, Mr. Pfefferland, next time you come, there will be a chicken barbecue for you. Thank you very much for your presentation. Mr. Walter and Mrs. Gorman, could you join us here?
We remember your lithium presentation. Questions have been received for the presentation. One is from Stefan Fischer. Stefan Fischer is asking, to what extent is the lifetime of the membrane defined already? Will it be clogged up or contaminated after a certain time of use? And do you know already whether the membrane itself can also be recycled? That's a very good question, of course. This membrane development has been taken forward very much in the last few months, and one major component is the powder or particle design of the membrane and then the external influence and impact. Here again, we are in a test phase in order to characterize what impurities or what conditions are unfavorable for the operation of the membrane and would shorten the lifetime. But we also have made major progress already, which will then also be reflected in the design of the electrochemical cell and in the way of how the specification of the water put in will be like, so as to increase the lifetime. And the second part of the question, the membrane. They do not have the lithium-ion conductivity anymore. They cannot be used anymore for the process. This cannot be directly recycled and be used to create a new membrane. You have to actually start again from scratch as far as the powder is concerned. So lithium is a topic that journalists are very much interested in. Another question from Gerald Henry from Chemical Engineering.
And for lithium recycling, will it be located in Hanau or in Marl?
Ja, das ist auch eine
That's another very good question. We are working on a laboratory scale right now. In Marl and in Hanau, we have laboratories in order to move forward in this field. And at both sites, we are developing a prototype concept. The colleagues at Hanau here, this is the major part of the engineering department with more expertise. And here we are one step further ahead with the building of the first prototype plant. How large it will be is difficult to tell right now, cannot be told right now. Another question is from Christa Friedl. She is asking, what were and what are the technical challenges in your process? How expensive will recycling be compared with the conventional processes? The challenges of the process, the ion-selective membrane electrolysis is a process that is very demanding, technically speaking. You really need membrane materials that are suitable for this application. That is the biggest challenge here. Stable membrane systems have to be created that are suitable for this job, to produce membranes. Compared with the conventional process, we have actually made first comparisons with respect to the operating costs, and we have come up with a very attractive cost situation, also in particular with respect to sustainability, because the CO2 footprint is much smaller and you consume a lot less water. And you have a process here that also has a high current efficiency. So this also gives an advantage in terms of operating costs. And then another question of Philip Broadwith for Chemistry World to you.
Recovering the other valuable materials from batteries.
Genau, das bin ich in meinem Vortrag nicht genau eingegangen. Ich sprach immer von Lithiumhalt.
I've always been talking about lithium-containing wastewater streams. They come from the leaching process flows or pre-process streams where cobalt and nickel have already been reprocessed and recovered, where lithium-containing waters are produced that are collected and then treated further. So with this technology, we are exclusively addressing the separation of lithium, but the cobalt-nickel recycling of the other biggest and or most valuable materials is untouched by this, is not concerned by this. So next time when you're here, we will take you home by an electric car. Thank you very much for participating. This brings us to Mr. Volter. This is the first question from Christa Friedl for you. She's asking, are the bacteria genetically changed? No, our bacteria have not been changed genetically. We have taken a different approach as far as this is concerned. We do not actually train properties on the bacteria, but we were looking for bacteria that have learned to fix nitrogen by nature. Therefore, we actually collected the bacteria with this in mind, and from the over 400 that we collected, we've picked the ones that are most suitable, that actually learned how to fix nitrogen by nature. Another question, how much different strains of bacteria do we actually use? We collected over 400, as I mentioned before. We do not just have one microorganism, we have 3 microorganisms in a combination in our product, and this combination provides the optimum of nitrogen fixation. Excuse me, Mr. Scott, there is another question of Alex Scott. Good that you are still here.
That black mass will be exported to China or to South Korea?
Wird das auch exportiert nach Südkorea oder wollen die auch Recycling ins Leben rufen? The biggest battery capacity is in Asia. Europe is in the process of building this up, so it may well be that these kinds of scenarios will take place, that black matter is actually exported. But I'm confident that in Europe, given this rise in mobility, rising trend of mobility and the building up of many battery factories on a giga scale, and then there will be also recycling companies like Unico and others that are building up new capacities and have already provided information that they can actually also reprocess the black matter, the black mass themselves. And this would actually foster the industry here in Europe. And this brings us back to Mr. Volta. Mr. Kiel is asking, so the plants are growing and forming new leaves. How often do you have to apply this bacteria solution, or does it make any difference if you just apply it once? Will that be enough? In our experiments, in our greenhouse trials, we applied it only once. We are now in the field trials, so we cannot actually judge this conclusively. But our hope is, on the one hand, that one application will be enough. But what's good about the application to leaves, unlike the conventional nitrogen fertilizer, is that I can actually apply it several times and can reach the plant. If you actually spread nitrogen fertilizer, you reach the leaves and it doesn't get to the roots and you lose more nitrogen, therefore you don't do it normally. And in our case, if need be, we could also re-fertilize. Then another question from the chat, bacteria are also living organisms. For how long can they be stored, the biostimulants? How long the bacteria can be stored? In our formulation, they are stable and have a long storage life. In the unique combination of the breakthrough and the cypionate with a protective layer that will dissolve. In our storage tests, we haven't reached an end point in order to find out when the end of the storage life has been reached. So we haven't reached the end yet. Let me look to the room again. Are there any questions here from the attending participants here in the room? There are satisfying smiles, and from this I conclude there are no more questions from inside the room. Thank you very much to the two of you. And now, Mr. Schwager, Mr. Marquardt, please, because one question has been received from Mr. Roybold. First of all, Mr. Pfefferle, in his presentation, mentioned Singapore twice. This brings us to the question, how important is the location of Germany and how important are other locations in the United States or in Asia for us?
Now, when you look at history, you see that research and development side Germany was the heart and core of the company. And obviously, with all the further developments over the past decades and in our process to become a global company on the basis of the development phases, applications, near customers, we have grown into other areas, further development into special chemistry, then further takeovers. And in the process, research sites in Asia and North America were added. In the past years, we looked specifically for topics in other countries, and we were able to identify the main topics for North America and Asia. And this is why we are focusing more and more on these regions as well, because applications in these other regions as well require a different focus. But nevertheless, Germany, Europe is still our center, so to speak, with the declared goal that in revenue growth in the regions outside Europe, we want to engage more and more in research and development application technology. And this is how we defined our locations. The one was mentioned already, the Singapore one, which was considerably enhanced in the past few years, then Innovation Center in Shanghai, and in the United States, recently in Allentown, we were able to inaugurate our new research and development site. Mr. Marquardt, in his new role, was able to cut the ribbon and look at the new site. Mr. Marquardt, your new role, you're taking over the office in difficult times. A lot of people are talking about a crisis. We have numerous crises our industry is confronted with. Do you feel that with your research and development activities you would have to accelerate things a little bit, save costs, for example? Well, crises are always opportunities for innovation. I was able to observe that when I had different roles in the group. Every time when there was a crisis, we were able to see kind of momentum, namely that customers and markets asked more and more for our innovations. So I'm pretty confident when I'm saying that crisis will lead to a situation where we come up as a solution provider for many of those problems occurring in a crisis. We'll always get an opportunity to prove to our customers that we have mature solutions available. And I experienced in the past that crises in part helped accelerate solutions. So there are disadvantages and advantages in a crisis. For innovation is an accelerator. Well, you have to grab your chance and your opportunity. I'm pretty confident when I'm saying we will manage. Mr. Schwager, are you sharing his confidence? Oh, definitely, definitely. As a company, we're able to prove that in the last years we kept our research level higher than the average in Germany, and we were able to prove that we make new research funds available, and we are willing to commission new research and development sites as well. So we've been successful in doing so, and this is translated in the growth in our innovation fields. And Mr. Marquardt will certainly have the pleasure to come up with new impulses as far as contents are concerned. Mr. Roybal from Chem Manager, since we're talking about numbers and figures, asking, could you tell us what the current contributions of innovation growth fields to the group results are? What kind of fields receive most of the investments at the time, and what are the fields who would be the biggest contributors until 2025. Thank you very much for your curiosity. I would like to satisfy it, but since as a group we are exposed to competition, we cannot give you any detailed information. As I said in my presentation, it is an essential contribution that stems from lipids. I can't give you any precise numbers because this would be comparing to competition. But there's something I can talk about because you were asking for investments. We are just about to look into our polyimide 12 complex in Marl, CDT, and ends up with CR12 powder. And this is to be integrated in the innovation growth field, and we are about to commission that plant. And you reported on the numbers. They are rather important. And this is part of the innovation growth field of additive manufacturing. And you were able to read in the media, and we reported on it, that especially when it comes to our membranes in the Austrian science, we have made investments and we have commissioned new plants. So we moved well ahead. It is not only on a small scale that we are active. No, we are active in a large scale, and this is why we are reaching the billion. There's another question by Mr. Kemp. The CIO, is it directly linked with the venture capital? What is the role of the venture capital in research and development, or are there two separate units? No, this is a clearly fully integrated thing. The venture capital, the Evonik venture as it is part of the research and development organization. GNI, excuse me, but also on the operating side, the question in which units are we going to invest. So close exchange, we see hundreds of companies per year which we look at, and then we'll pick out the ones that are the best match with our research fields. Let's take Walter Pfefferle's presentation, Epigenetics and the chicken. There's another ethical problem. There is the ethical problem that the male chickens directly after hatching will have to be killed. Now, the question is, how can you solve this ethical problem? And this is how in Evonik Venture Capital we participated in a startup in Open where the determination of the gender basically can be done prior to hatching. This is the way we tackled the issue, so we're moving ahead. There's research activities going on. And first analysis and devices have been carried out. It's not only on a laboratory scale, so we're in a reality scale already. Or a different topic, let me take an example from additive manufacturing. We are very deliberately working with powders, but via Evonik Venture Capital, we were able to get in touch with a startup in the United States where we took a share, and at the end we took it over. Structured polymers is the name. They were not working on the level of powders, but rather on a level of microgranulars. This is how we broaden the application options of this technology, moving towards new products, new polymers, because we are mainly working with PA12 powders. And this is why we always need link between research development, operating business, and the competence of a venture capital link, which will look into seed financing, which is experienced in seed financing and can harvest all the opportunities from there. And this is exactly the way we are going to deal with Sustainable Tech Fund, namely full integration in the research and development landscape. 2 weeks ago, Mr. Marquardt, we were together in the Research and Development Council. And this is where Evonik Venture Capital is an integral part. The director, Bernard Moore, was present in this meeting, and we took sufficient time in order to look into all the Evonik Venture Capital topics. Mr. Marquardt, Mr. Schwager, thank you very much, dear journalists. Thank you very much for participation. Thomas Schmidt, if you hear me, you've got one question to Mr. Walter. You hand it in. We'll call you back later on, and you will get an answer to your question. So I would like to look around. Is there anybody in the room who would like to take the floor? This doesn't seem to be the case. Mr. Schwager, Mr. Marquardt, technicians, interpreters, camera, thank you to all of you. I close the press conference. I'm pleased to see you.